हेलो फ्रेंड्स, वेलकम बैक टू माय यूट्यूब चैनल। इस वीडियो में हम एमएससी जूलॉजी फोर्थ सेमेस्टर का फर्स्ट पेपर न्यूरोफिजियोलॉजी एंड जनरल फिजियोलॉजी सब्जेक्ट का स्ट्रक्चर ऑफ सैक्रोप्लाज्मिक रेटिकुलम एंड इट्स रोल इन मसल कॉन्ट्रैक्शन का स्टडी करेंगे, जिसका इंट्रोडक्शन है। मसल कॉन्ट्रैक्शन इज ए वाइटल फिजियोलॉजी प्रोसेस दैट इनेबल मूवमेंट इन ऑर्गेनिज्म. Understanding the mechanism behind muscles contraction, including the role of specific cellular structure and enzymes, is crucial for comprehending how muscle functions. This essay will explore the structure of the sacroplasmic reticulum and its role in muscle contraction, the various theories explaining muscle contractions, the physiological changes and that occurs during contraction, and the enzymes involved. The discussions will culminate with a conclusion and a reference to help further study the topic. Definitions of muscle contractions. Muscle contractions refers to the process by which muscle fibers shorten in response to stimuli leading to movement. This contraction is driven by a complex interaction between actin and myosin filament within the muscle fibers. In skeletal muscles, the contraction is regulated by the nervous system, with the sacroplasmic reticulum playing a central role in calcium ion regulation. Structure of the sacroplasmic reticulum. The sacroplasmic reticulum is a specialized type of endoplasmic reticulum found in muscle cell. It is extensive network of membrane-bound tubules and sac that surround each myofibril, the contractile unit of muscle fiber. The SR serve as the primary intracellular store to calcium ion, which are essential for muscle contraction. The sacroplasmic reticulum consists two main regions. First, terminal cisternae and second, longitudinal tubules. First, terminal cisternae. These are enlarged section of the sacroplasmic reticulum located at the junction between A and I bands of the sacromere, the functional unit of the muscles. They store large amount of calcium ion. Second, longitudinal tubules. These are less dilated portions of the sacroplasmic reticulum that run parallel to myofibrils. They facilitate the transport of calcium ion throughout the muscle fiber. Role of sacroplasmic reticulum in muscle contraction. The sacroplasmic reticulum play a critical role in muscle contraction by regulating calcium ion level in the muscle cells. When an action potential electrical signals travel along the muscle fibers membrane, sacrolemma is triggered. The release of calcium ion from the SR sacroplasmic reticulum into the cytoplasm. This calcium ion bind to troponin, a regulatory protein on the actin filament. This binding causes a conformational change in tropomyosin, another protein, which exposes binding sites on actin for the myosin heads. The interaction between actin and myosin results in the sliding filament mechanism of muscle contractions. After contractions, the calcium ion are actively pumped back in to the SR through the sacroplasmic reticulum calcium ATPase pump, allowing the muscles to relax. Theories of muscle contractions. Several theories have been proposed to explain the mechanism of muscle contraction. The two most prominent are the sliding filament theory and the excitation contraction coupling theory. First, sliding filament theory. This is the most widely accepted model. This suggests that muscle contraction occurs when the thin filament actin slides past the thick filament myosin, causing the sarcomere to shorten. This movement is driven by the energy released from ATP hydrolysis by myosin heads. Second, excitation contraction coupling theory. This theory explains process by which an action potential leads to muscle contraction. It involves the sequence of events where the electrical signal from the motor neuron is translated into a mechanical response by the muscle fiber. The action potential triggered the release of calcium from the sacroplasmic reticulum, initiating contraction through the sliding filament mechanism. Physiology of muscle contractions. The physiology of muscle contraction is a step-by-step process involving electrical signal and biochemical reactions. First, neuromuscular junctions. The process begins when a motor neuron releases acetylcholine at the neuromuscular junctions, which depolarize the muscle fiber membrane. Second, action potential. The depolarization triggers an action potential that travels along the sacroilioma and down the T-tubules, which are extensions of the sacroilioma that penetrate the muscle fiber. Third, calcium release. The action potential reaches the terminal cisternae of the sacroplasmic reticulum, causing the release of calcium ion into the cytoplasm. Fourth, contractions. Calcium ion bind to troponin, moving tropomyosin and exposing the binding sites on actin or myosin. Myosin heads energized by ATP from cross bridges with actin and pull the thin filament toward the center of the sarcomere, causing contractions. Fifth, relaxations. When the action potential cases, calcium is pumped back into the sacroplasmic reticulum and the muscle relax. Changes during muscle contractions. During muscle contraction, several changes occur at the muscular and structural level. First, sacromere shortening. The distance between Z line decreases as the actin filament slides over the myosin filament. The I band actin shorten while the A band myosin remains the same length. Second, cross bridge formation. Myosin heads to bind to actin, forming cross bridges. These cross bridges pivot and pull the actin filament toward the center of the sarcomere. Third, ATP consumptions. ATP is required for the myosin heads to detach from the actin and reset for another contraction cycle. Enzymes involved in muscle contractions. Several enzymes are critical for the biochemical processes involved in muscle contraction. First, ATPase. ATPase is present in the myosin heads, where it hydrolyzes ATP to provide the energy necessary for muscle contractions. Second, sacroplasmic reticulum calcium ATPase. This enzyme actively pumps calcium ion back into the sacroplasmic reticulum during muscle relaxation. Third, acetylcholine esterase. This enzyme breaks down acetylcholine in the synaptic cleft, stopping the action potential transmission and allowing the muscle to relax. Conclusion. Muscle contraction is complex, highly regulated process that essential for movement in animal. The sacroplasmic reticulum play a central role in regulating calcium ion level, which are crucial for sliding filament mechanism. The theories of muscle contraction, particularly the sliding filament and excitation contraction coupling theories, provide a framework for understanding this process. The physiology of muscle contraction involves a series of steps, from the release of acetylcholine to the return of calcium ion to the sacroplasmic reticulum. Several enzymes including ATPase and SCRCA are involved in energy production and muscle relaxations. Overall, muscle contraction is a finely tuned process that allows organism to perform a wide variety of movement. References: